about some more information about DNA and genes and what DNA does within the cell. So our goal is for today to talk a little bit about DNA structure and how it's bonding and what its capabilities are, and how that structure impacts its function. It's kind of a theme that we've been talking about, is how the structure of certain biomolecules impact their function. We're going to talk about how DNA gets replicated within the cell. We're going to talk about what's called the central dogma of biology, which central dogma code for a very important part of biology. And we're going to kind of flesh out each of the individual stages in that expression. And we're going to be talking about the different phases and enzymes involved in both transcription and translation, which we'll be introducing today. Okay, so DNA, we've kind of described it as like blueprints of the cell, the instruction manual of the cell. What it does is it stores information for synthesizing proteins. So here are some of the different functions of proteins. So proteins can be used to do a wide array of things. So nucleotide metabolism, so DNA is a nucleotide, so making larger nucleotides is one of the major functions of proteins, which is kind of interesting because DNA directs the production of proteins, then proteins come back and make more DNA. We have cell growth and maintenance, protein metabolism, energy metabolism, so that's like cellular respiration and fermentation, things like that. Transport, so moving things around. Apoptosis is a major category. So apoptosis is what again? Cell death. Great. And then this signal transduction is a huge part of proteins' functions. Does anyone want to make a guess at what that is? What could signal transduction mean?
Sending signals through cells, DNA.
Yeah, so that's pretty close. So basically what a cell is constantly doing is monitoring its external environment, and information from that external environment has to get passed into the cell. And so it does that with proteins. So there's pathways of proteins that sense the external environment, react in some way, and then pass that signal back into the cell to change some sort of cellular behavior. So proteins do a wide array of things for the cell: sensing the external environment, making larger biomolecules, moving things around, directing the cell through cell death. All of these things are mediated by proteins. And so protein production is guided by DNA. So this is the central dogma of biology. So what this is saying is that information goes from DNA to RNA, which is a type of biomolecule that we'll talk about more on Thursday. And that information goes into making proteins. So DNA, just going to stay in the nucleus, is sending information out to make proteins. So the process of making more DNA is called replication. So this happens during S phase and interphase. It's replication. Transcription is the process of making a copy of the DNA in the form of RNA, and that's going to leave the nucleus. So the DNA never has to leave the nucleus. It makes a copy of RNA that it sends outside, and then that gets translated into another form of biomolecule called proteins. So we have information in the nucleus in the form of DNA. We make a copy of RNA that we send out of the nucleus, and then that RNA gets translated into proteins. And proteins have functions in the cell to change cellular behavior and to sense the external environment.
So I think you said that the RNA is just like a little bit of DNA, or?
Yeah, so the DNA in our cells, we talked about this last week, is just a very massive amount of information, a very huge biomolecule. The RNA is the relevant portion of that DNA that's needed to make a specific protein or set of proteins. So it's like a smaller, more manageable copy that's like less valuable. Because we never want to, we talked a lot about how the DNA gets sorted and how there's all this regulation of the cell cycle to make sure nothing really gets messed up in the cell in terms of DNA, so everything can stay in sync, basically. So all of our cells are behaving the same way. And so one way to preserve that integrity of the DNA is to make sure it only stays in the nucleus and it never leaves. And we use RNA as a way to take information from the DNA and move it out of the nucleus in a way that isn't as valuable. So if this RNA gets chopped up and degraded by enzymes in the cytoplasm of the cell, which is very common. Things are usually getting degraded in the cytoplasm. Then it doesn't really harm the cell that much because it's just a copy of the DNA. It's not like the real thing. So chapter 12, we're going to be talking about these 3 steps. So today we're going to talk more about what DNA is, and then talk about replication, and then Thursday we'll talk about transcription and translation. Any questions about what we're doing or why this is relevant? What? 12. Yeah.
Does the cell make the DNA from the RNA like out of nothing, or what is the purpose of eating protein?
Of what?
Eating, ingesting protein. Protein and then change the protein?
Yeah, so that's actually, so we eat protein, and those are going to be long polypeptides, like structures of amino acids. And as we eat it, it gets digested and broken up into individual amino acids, more or less. And what our cells do is they take those individual amino acids, they listen to the instructions from the DNA, and they make the proteins that we need at that moment. So like if you had a cell that needed to go under apoptosis, it would take amino acids from the bloodstream that you digested and make the right proteins so it could undergo apoptosis, and then it would.
So it makes its own primary structure with like little pieces.
Exactly. Yeah. Great. And we'll dive into that more in the translation chapter, section. But you're on the right track. So DNA has a lot to do in the cell. It's a very important biomolecule. So there's really 3 important characteristics that we know DNA has to have. Just because we know what its job is, so we know what capabilities it has to have in order to fulfill its role. So it has to be capable of storing information in somehow, in some capacity. So DNA has to be directing protein synthesis. If it's not doing that, it's really not serving much purpose in the cell at all. So it has to be capable of storing information. It has to be capable of being replicated with very high accuracy. So each time we go through S phase of interphase, we need to be making basically exact copies of our DNA. But we also have to have a small amount of variance as we undergo interphase. So we're always going to want to be inserting a small amount of genetic diversity each time we're undergoing this DNA replication, because as we increase the variability, we kind of increase the possibility that as a population as a whole, that we can adapt to changing environments. So this is another way that biology increases genetic diversity. So this is something that we saw a few times in the previous chapter when we talked about crossing over, independent assortment, sexual reproduction, and the error rate of DNA replication is another way that the cell increases genetic diversity. So these are the 3 things we're going to keep in mind as we talk more about DNA's actual structure. So DNA is made up of 3 main groups. So we have our phosphate group, we have a sugar, which in DNA is deoxyribose is the sugar. So I mentioned RNA earlier. RNA has a different sugar. That's actually just called a ribose. So that's the difference between DNA and RNA is the sugar, and there's also another difference that we'll dive into a little bit more later. And also there's this base up here. Thank you. So this base is different between different nucleotides. And the order of the bases is how the DNA is going to be storing information. Thank you. So we have these long stretches of DNA, and there's 4 different possible bases that DNA can have. And the order of those individual bases is how the DNA is going to be storing information. So that's both like a change in the structure of the DNA, and it's like a change in just purely the information within the DNA. So what I mean by that is there's 4 different bases, so there's many different possible combinations that DNA can have, but as it changes those combinations, we're also literally changing the structure of the DNA. So it's going to change the way that it interacts with other chemical forces, and the way it interacts with those different chemical forces is how that information is going to be transmitted. So it's sort of like two levels of information. It's not just the order that can be like read out by other biomolecules. It's also changes the way that the DNA behaves in different chemical environments. Does that make sense? I think it'll make a little more sense as we keep going. But the DNA is going to respond differently based on what type of base that it has, and that's how we're going to use it to transmit information. So again, there's 4 different bases, and so for humans, each chromosome has approximately 140 million base pairs. So we'll talk more about what base pairs means, but this number 140 million, this is a massive amount of bases. And that's why, we only have 4 different possible bases, but with 140 million bases in the single chromosome, we can really store a lot of information.
Within DNA, you said that there are different genetic sequences of alleles,
Yeah.
traits and such. If say one of them was like ACGT, could there be another section in that same DNA that's also ACGT for like a different trait? Because to me, when I think of that, it'd be like 2 of the same traits repeating, but could it be translated into something different?
Right. Yeah. So the way it gets processed from the DNA could be different for different genes. So that's going to change. Even though the DNA could have the same sequence, it could have different instructions on how that gets processed as the RNA leaves the nucleus to make proteins. So it's not only what's on the DNA, it's also how that gets copied and processed into making proteins. I think this will make more sense as we keep going, but let me know by the end of the class if that doesn't make sense. So these are our 4 bases in DNA. So again, DNA, we have 3 groups. We have the phosphate and the sugar, which are the same for all bases, and then we have this base that's varies. There's 4 different combinations. So we have an A, a G, a T, and a C. You don't have to memorize guanine, you can just remember G. Because honestly, until we come back to this chapter, I always forget each year. Because really all you need to know is the letter, and that'll get you through basically any application for DNA that you need. So what are some things that we recognize about these different bases? How are they different? Or how are they similar? Anything that kind of sticks out? Any themes? Yeah, so the size of them, maybe. So we have 2 rings here and a single ring here. Any other differences that we see?
I see a similarity when they all have E.
Yeah, they all have these nitrogen groups. Yeah, good. Did someone else say something similar? Yeah. So what happens? So N nitrogen is very electronegative, and hydrogen, is that electronegative or not? It's a very low electronegativity. So what happens when we have an N and an H bonded together? What could happen there? Yeah. So when the N is bonded to the H, we're going to have a lot of the electrons over near the N and very little near the H. So this will be partially positive, this will be partially negative. And then we have some oxygen groups here. These will be partially negative. So we have a lot of opportunities for hydrogen bonding between these bases. So we have different sizes. We have sort of similar compositions where we have these nitrogens and hydrogens bonded together. We have some opportunities for hydrogen bonding. So we're going to talk a little bit more about how the hydrogen bonding and the size of these bases influences their behavior. So DNA, when it's in our cells and it's kind of like a healthy configuration, forms this what's called a double helix. So we have 2 strands of DNA bonded together here. So along what's called the sugar phosphate backbone here, we have where the sugar and the phosphate are bonding together. That's going to be through covalent bonding. And then between bases, we have hydrogen bonds. So here's the sugar phosphate backbone with covalent bonds. And between bases, we have hydrogen bonds. So hydrogen bonds are a lot weaker than covalent bonds. So we have a very strong bonding structure along this backbone That's going to keep the DNA intact. And then we have these hydrogen bonds, which are a lot weaker, and it's going to make it easier to pull them apart down the middle like this. But we have this double helix structure with our two strands of DNA that are bonded together. Has anyone heard the term double helix before? Yeah. You can write about that in your paper if you want, which is going to be a really fun experience, I promise you. So we have these double ring structures and the single rings. So you see, okay, so in order for the structure to remain strong and intact all the time, we can only allow, or the cell only allows bonding in which we'll have the same width of our rungs of our ladder. So to make that happen, we have only bonding between double rings and a single ring. So we have two rings in these spaces, and a single ring and a single ring. So we're always going to have in our DNA a double ring base bonding to a single ring base. So that means we're never going to have A and G or A and A bonding together. And if we also look at the hydrogen bonds, so A and T both make two bonds. So why don't I write this on the board? So we have number of rings, 1 or 2. Number of hydrogen bonds, 2 or 3. All right, so which base that only has one ring can make two hydrogen bonds? If we look up here, we can see A has two rings and makes two hydrogen bonds. T has one ring and makes two hydrogen bonds. C has one ring and makes three hydrogen bonds. So let's fill this chart out. So what's
T and then A.
So it has one ring and two hydrogen bonds is which one? T.
And then A is 2, 2.
Does everyone see how we're doing this? You all. So T has one ring and two hydrogen bonds. A has two rings. It's not a very good ring. Wow, terrible. And two hydrogen bonds. And so what base has one ring and three hydrogen bonds? A little hard to see, isn't it, from there? So we can see the hydrogen bonds. Let's see, we've done T, so T has these two spaces to make hydrogen bonds here. Actually, it would be here and here. But then, if we wanted to find one that has one ring, you can make three hydrogen bonds. So it would be one of these, either C or G. So which one of those is one ring and three hydrogen bonds? Yeah, so C is one ring and three hydrogen bonds, and that leaves G. So now we're going to talk a little bit about what this means for base pairing. Hopefully, I will learn how to make double ring at some point. So if we have to only bond between nucleotides that we're only bonding between a single ring and a double ring, and they have to have the same number of hydrogen bonds, what are the possible combinations we can have of bases? So, yeah, there's two possible combinations. So, like, T and C couldn't bond together because that would be two single rings and it would be too narrow. and that would put stress on our sugar phosphate backbone, and those wouldn't have a very good connection. And also, we wouldn't really be fulfilling C's need to maximize its hydrogen bonding, so it wouldn't be as stable. So
maybe before technically order matters, right? Because TA and A2.
That's a great point. That's not considered order at this point, but I see what you're saying. So A and T can bond together, and G and C can bond together. But you're right, in the future, order will matter. But if we're just talking about what can bond together, this is how we would figure it out. So the point of this is to show that if we know what one of these strands is, so if I only could figure out what these bases on the left side were, since I know how the bases bond together, I could figure out exactly what's on the right side with no error, with exact certainty, basically. That's because the way that nucleotides bond together is very predictable because of their structure. And that gives them a function that makes DNA replication very easy for the cell to do. Because all you need to know is one side of the DNA, and then you know exactly what the other side is. So it makes it much easier for the cell to replicate the DNA. Were there other questions? I feel like I was talking over someone. Yeah.
On there, there's the A, T, G, and C in a little box where it shows all the different elements.
Yeah.
Where are they connecting? Where are the hydrogen bonds?
Yeah, that's it. Okay. So we'll get hydrogen bonding between this NH will create a partially positive hydrogen, and this O will be negative. And then this O would also be negative, but it wouldn't make a hydrogen bond, I think, just because of its orientation. So you'd probably get bonding here and here. With the A, you would get bonding, I think, just here and here.
Oh, so the picture over there is a little weird.
Yeah, they're not, because like this would be oriented around like this, so it's not totally clear. Or maybe it wouldn't be here, because that would be a carbon and hydrogen bonded. Yeah, I'm not totally sure. It kind of depends on the way it'd be oriented on the nucleotide.
It makes it look like it's on the hexagon part of the A with the T on the bottom.
Yeah.
That's not necessarily where they are. That's just like.
Well, I mean, I could be reading it incorrectly, to be honest with you. So yeah, it does look like it's on this bigger 6 carbon ring here that has the bonds. And so the black, this must be the hydrogen, and this must be a negative region here. So it would probably be like here. No. I'm not totally sure. That's a good question. Sorry. That's something I can look into a little further before the next class. But the important thing that I want us to take away from this is that based on knowing the structure of the nucleotides, we can determine how they're going to bond together, and we can figure out that all you need to know the entire composition of a chromosome is what half of the DNA is, what one of the strands is. So I know what this left strand is, I know exactly what the right strand will be. And the cell is going to kind of exploit that to make replication easier. And so that phenomenon is called complementary base pairing. So complementary base pairing is the set of rules that tells us that A is always going to be bonded to T and G will always be bonded to C. And so one result of that is that if we look at the genetic composition of different species, we'll see that the amount of A is always roughly equal to the amount of T, while the amount of G is always going to be roughly equal to the amount of C. So if we go through and sequence all of the DNA in these different species, we can see that they're following this complementary base pairing. And they're doing this because it's necessary for the DNA to stay intact. So complementary base pairing arises from the structure of the nucleotides. The cells use it to make replication easier. And we can see evidence of it when we sequence, or we look through, we use different forms of technology to look through what the composition of the DNAs in different species. That the amount of A in the chromosomes is roughly equal to the amount of T, the amount of G is roughly equal to the amount of C. And this is a result of the complementary base pairing. Oh, sorry, this is the figure that you wanted. Yeah.
I think the black dots were negative.
Yeah, I think I might have been wrong then. Well, there we go. I should put this on the previous slide and like see what is what. So yeah, that was maybe close. I'm not sure. All right, so that's complementary base pairing, and that arises from the number of rings, the number of hydrogen bonds that the nucleotides can make. So we talked a little bit about this double helix. So again, we have two strands of DNA that are kind of wrapped around like a spiral staircase like this, like the two railings of a spiral staircase or like a double helix. That's like the overall three-dimensional structure of the DNA is a double helix. And now each strand, we say, is anti-parallel to the other strand. So has anyone heard the term anti-parallel before? It's not the term that I would have used to describe DNA, but it's a term that's very well accepted, and so we have to kind of adapt to that. So if we look at DNA, we have these sugar and phosphate groups, and we call the phosphate group the 5' end, and we call the sugar group the 3' end. And so a lot of cellular machinery will only run one direction, either 5' to 3' or 3' to 5'. And so this orientation of the strands is going to matter a lot when we talk about how different enzymes interact with the DNA. So we have one strand that has like the phosphate group on top and the sugar group on the bottom. If we were to use like a top and bottom orientation to look at the DNA, and we'd say that runs 5' to 3'. But the strand that it's bonded to is parallel to it because they're in parallel orientations, but it runs the opposite direction. So it goes 5' to 3' this way from bottom to top. So that means if we have some machinery that only runs 5' to 3', it's going to go this way on this strand and that way on this strand. So it's going to add a little bit of complexity when we talk about how we're going to be copying the DNA and reading it off to do other tasks. So we have a double helix, these two parallel strands that are twisting around each other, but each strand is what we call anti-parallel because they're parallel to each other. You know, they're not going to intersect, they're not going to cross over, but they're running in opposite directions. It's kind of like, you could say like lanes on a road are anti-parallel. They hopefully are not going to cross. That would not be ideal, but they're running in opposite directions.
What's running?
It's really just the orientation. So it's just like for this base, it has the phosphate on top, and this base has the phosphate on the bottom.
Right.
But when something attaches to it, like if we had an enzyme attached to this base, it would want to go 5' to 3', so it would want to go this way. If we had an enzyme attached to this base, it would want to go like that way. It'll make a little more sense as we get going, but we're talking a little bit about the different structures of DNA. So we know how nucleotides bond together, and now we know how strands of DNA are going to orient themselves in relation to each other, and what the general three-dimensional structure is. All right, let's take a short break. You want to get in groups of like 2 to 4, and we'll talk about these questions. Feel free to jot down whatever you talk about, and we'll get back together in like 5 minutes and recap what we learned. Feel free to move the desks. Be excited and energetic. Don't leave. It's going to get better. We're going to finish it today. So, DNA replication, we've talked a little bit about before when we talked about interphase. So during the S phase and interphase, the cell was replicating the DNA. So it was making an extra copy of all of our DNA so that when we undergo mitosis or meiosis, that will have enough DNA to make all of the diploid or haploid cells at the end. So during S phase of interphase, we're copying our DNA, and that process is called replication, and that's what we're going to be talking about today. So the goal at the end of DNA replication is to make 2 identical copies of the DNA. So that's what the goal is. And now it's going to do this through a process called semi-conservative replication. So who wants to kind of paraphrase that term to me? Semi-conservative replication. Yeah, basically. I don't want to. It's what? What do you mean?
It replicates some stuff.
Right. So it uses one of the strands as a template and adds on to that. So basically what it's going to do is it's going to split our DNA in half along the hydrogen bonds because it's easy to pull those apart. And it's going to use each of those strands as a template strand and add the appropriate nucleotides based on complementary base pairing to those template strands to create 2 identical copies. And remember, since nucleotides have such a restricted binding behavior, that if we know one strand, we know exactly what the other strand should be. So by splitting it in half, we can use each of those strands as a template strand, and at the end we'll have exactly identical copies of the DNA.
Enzyme, like you were talking about before, that you run down one side and copy stuff?
Yeah, it's a series of enzymes that are going to pull it apart, copy it, and proofread it, basically. But yeah, you're on the right track. So this is semi-conservative, is we're going to split it in half and use the existing strands as template strands and add on to that. So we're conserving what was there initially, but we're also adding on to it to make the new strands. So we're conserving what was there before, but we're also adding on to it, so we call it like semi-conservative.
Are they always in strands
Is
there a number?
it's usually like many strands, like millions of base pairs. So 2 strands of like millions of base pairs. Yeah. Can I go on? Are you still? So the 4 main steps to replication, we're going to unwind the DNA. So this is going to be like twisting it out of this double helix and separating the strands, this unwinding step. We're going to be undergoing complementary base pairing on these separated strands. So we're going to be adding on the appropriate nucleotides to the template strand to make our new strands. We're going to be joining together our DNA, so to create a cohesive strand again, rather than just like separate fragments. And then we're going to be proofreading the new strands to make sure there's a very low error rate. So these are the 4 steps that we'll get into more detail on. Do we see how this is kind of reaching our initial goal of copying our DNA during S phase and interphase before we split the cells? And remember, this is all happening in a very tiny volume for a very huge amount of DNA. You can really think about DNA as like a zipper that always wants to be zipped shut. So what we do is we have an enzyme, DNA helicase, that's going to come in and unzip the DNA. And so that's an active process that requires like an investment of energy. So if this is an endergonic process, we have to couple it with another exergonic process to make it happen, right? And so we're going to use an enzyme to make sure that our activation energy is low, but we still have to be expending energy in the form of ATP to make all of this happen. So we have an enzyme, DNA helicase, that comes in, and as we add in ATP, it unzips and separates the individual strands of the DNA. And then we have other proteins that come in, single-stranded binding proteins that come in and prevent it from shutting back together. Because if we unzipped it and it just closed back again, then we would just be wasting energy and we wouldn't be able to get other enzymes in there to copy the DNA. So we have a DNA helicase that comes in, separates the bases, and unwinds and unzips the DNA. And then we have our single-stranded binding proteins that come in and stick to the DNA to prevent them from closing in on each other again and rebonding. Because if they just closed in on each other again, then we wouldn't be able to get other enzymes in there to copy the DNA. So after we unzip our DNA and we added these single-stranded binding proteins, we're going to have another enzyme called DNA polymerase come in. And that's going to be kind of facilitating this complementary base pairing. So we have these individual strands that we've broken apart that each have nucleotides sticking out. And now we want to come in and add the appropriate nucleotides to make another double-stranded copy of the DNA. So the DNA polymerase comes in, and it's going to add these nucleotides and create covalent bonds along the sugar phosphate backbone. And this is another enzyme that's going to be driven by ATP. So we're going to have this enzyme here with ATP coming in constantly, and these new nucleotides to copy the DNA, to undergo this semi-conservative replication. So DNA polymerase is one of the enzymes that's involved in the complementary base pairing phase of DNA replication. And we also have this DNA primase that is involved. So DNA polymerase only works from existing strands. So let's see. This is our DNA. DNA primase is going to come in and add what's called an RNA primer. So this is the RNA primer. This is our DNA. And once this is here, now our polymerase can attach. But what can happen, like cannot happen, is the polymerase attaching just to a single strand. I lost one of my caps. How did that? Okay. So the DNA primase adds a primer that the polymerase can work from, because the polymerase can't just attach to one of these single strands by itself. So our polymerase can't come in here and just start doing its thing. It has to be attached to a primer, like here. So here we have the DNA primase that makes these RNA primers that the polymerase can then attach to and start working from. But the polymerase cannot just attach to a single strand like this. So after we've separated our DNA with the helicase and we've kept it separated with the single-stranded binding proteins, then we have to have the primase come in, lay down an RNA primer, and again we'll get more into what RNA is later. And then once that primer is there, then the DNA polymerase can come in and start going to work.
So this DNA strand is like super, super long.
Yeah.
And it just rips apart right from the center. Start down at the very end.
No, so it does start from the center, because if it's
So it would be like an open loop.
yeah. So we'll why don't I draw that as well? So really, what you have is these are called like replication bubbles. So if the black is like the template strands, you'll have individual bubbles where replication is all happening from, so it goes much quicker. Because if it just started from the end and had to go like 140 million long, it would take a very long time. But the cell is very clever, and it allowed all of this to happen at individual little locations, so it can go a lot faster. But we'll get a little bit more into that later, I think. Okay, I'm just going to run through the next two steps kind of quickly, and we'll have to review them next class, but I just want to kind of show you the whole picture before we leave. So one, we talked a little bit about this like anti-parallel structure of the DNA. One kind of result of that is DNA polymerase only runs in this 5' to 3' direction. So on this strand, what's called the leading strand, it's fine. It's just going to basically follow this helicase as the helicase separates the DNA. And the polymerase is just going to go in the same direction and copy everything into one long continuous strand, and it's pretty simple. This strand, unfortunately, is running the 3' to 5' direction. If we look at it in this direction. So the polymerase cannot just go this way. Polymerase would not fit on the DNA and move in that direction. It only moves in the 5' to 3' direction. So instead what happens is every time a new section gets exposed, the RNA primer is laid down, and the polymerase keeps going and going until it hits another RNA primer, and then it pops off. And this is called the lagging strand, and we have all these little individual fragments of DNA interspersed with primers in between them. And these fragments are called Okazaki fragments. So they're little fragments along the DNA on the lagging strand that then later another enzyme, DNA ligase, has to come in and fix all of those little fragments to make them a long continuous part of DNA. So since DNA is anti-parallel, we have a leading strand where polymerase just follows the helicase, and we have a lagging strand where polymerase has to go the opposite direction of the helicase. So the helicase is going this way down the chromosome through the DNA, separating the strands, and the polymerase on the lagging strand is going the other direction. And that creates all these little fragments in between that the cell has to go in later and fix with DNA ligase. And the final step that we'll cover more in depth next class is another form of DNA polymerase comes in and it fixes any errors that the cell made in the first place. Not all of them, but a large degree of them. So I think I remember reading before, there's like the first pass is like 1 in 10,000 bases is a mistake, but then after this proofreading step, or no. The first, yeah, I'm sorry, I misread it. No, the first pass, it's 1 in 100,000 base pairs is a mistake, and then after this proofreading step, it's like 1 in 100 million base pairs is a mistake. So it drastically reduces the error rate to undergo this proofreading step. All right, that was all I wanted to cover today. Thank you for bearing with me.